Good evening and welcome back. So we're in the early part of the course where we're thinking about scaling, scales and scaling. And tonight, what I want to do is I want to focus on a beautiful case study that I hope you'll find enlightening and interesting. And that is the scaling of the size of the nucleolus with the nuclear volume or the cell size, since those are related. Before we begin, I just want to remind us all to not take for granted these images that we see in electron microscopy. The one on the bottom is from this guy named Don Fawcett. He was a really amazing electron microscopist who, in the 1960s, put together these atlases, kind of following on the heels of people like Keith Porter and George Pilati. It's a beautiful era in modern biology, cell biology, in the 40s, 50s, when people were developing the use of the electron microscope as a tool to try and understand the structures of the cell. And I think what I want to say is I just want you to take a look at this picture and for a moment just, kind of imagine yourself as an alien coming and discovering life. You look at this and it's clearly got all sorts of wonderful and intriguing order associated with it. And what I want to express is both the structural things that we don't want to take for granted. So, for example, the mitochondria have this very interesting stereotype structure, as do the Golgi apparatus, but then also the functional consequences, which so much has been learned about. So, most of the things that I'm showing you here are so-called membrane membrane-bound organelles. That is to say that the organelle, such as the mitochondria, the ER, the Golgi, etc., the nucleus, they're bounded by a membrane. And when I say membrane, I mean for a lipid bilayer or a double lipid bilayer and so on. And one of the points that I wanted to make in this vignette is I wanted to comment on the fact that there are these interesting correlations. And cognizant of this notion that correlation does not imply causation, that's fine. What I do want to think about, I want to be cognizant of, is the idea that correlations often demand an explanation. They're a curiosity. They're something that we would want to think further about. So what you see on the top is the scaling of the size of the nucleus in POMB, which is a yeast cell. So these are different than the brewer's yeast that maybe some of you are used to, but this is also a key model organism. And what you see plotted on the right is the volume on the x-axis in microns cubed, and then on the on the y-axis is the nuclear volume. So that's a scaling relationship that's interesting. The middle panel is the scaling of the spindle, and I just remind you that that's the apparatus that segregates chromosomes. And in a beautiful series of experiments, there's several different papers in Science. One of them I remember is from Rebecca Heald's group and Dan Fletcher, and I think Matt Good was one of the lead authors on that. There's another one that maybe was even in the same issue of Science and very interesting to look at. And here I think, if I'm a little bit fuzzy, I haven't looked at this in a while, but what they had were droplets, and then the size of the droplet was something that they could control, and then they could measure the size of the spindle as a function of the diameter of the droplet. Another very cool thing. And then on the bottom left, the last one that I want to comment on, and this is in yeast. What you see is that the volume occupied by the mitochondria is also correlated nicely with the volume of the cell. So this is just by way of introduction to this topic of scaling of organelle size. And I want to say that just structure of organelles, how they adopt the structures that they do, it's mysterious. It's not an obvious thing. And then there's these extra insights that have to do, empirical insights that have to do with things like the scaling. So this paper is going to be the basis of what I talk about in the remainder of this vignette. It's a beautiful paper that was written by Steph Weber when she was a postdoc with Cliff Brangwyn, and I feel very happy to say this about Steph, which is both her grad work and her postdoc work were of the variety of what I often refer to as experiments to change your life for. And when I say those words, what I have in mind is really this impression, this feeling that I have often after visiting somewhere and talking to people about science all day, that's a joyous occasion. But the most joyous of all is when I get on the plane on the way home and I think to myself, wow, that was so cool. I wish that my lab members and I were working on that as well. This is one such paper. So in a nutshell, there's this emerging field over the last, say, 15 or so years of membraneless organelles. And this is a very interesting segue to connections with the world of physics, because really the words that are now used to describe this kind of phenomenon are the words phase separation. And hopefully later in the term we'll have an opportunity to talk about phase separation. If we don't, I encourage you to go look at last year's edition of this course. It all exists on YouTube. There's 150 vignettes, and I did a fairly careful vignette on the Flory-Huggins theory and how to understand why it is that two different molecular species could either mix or phase separate. That's a deep and interesting question. Here, for the moment, what we're going to do is we're going to examine a different point, which is we're going to try to understand the scaling of the nucleolar size with the nuclear volume. So the first experiment that I'm going to show you about is shown here. And what you see in part A on the top is the C. elegans embryo. I remind you, C. elegans is a key model organism. It's a nematode. This is in the embryonic development. And what you see is the 8-cell, 16-cell, 32-cell, and 64-cell stage. Note that this occurs at fixed overall volume, and basically the words I might use for that is that you have reductive cell divisions. Every time there's a cell division, the volume of the cells decreases. And then what's plotted on the bottom, or not plotted but shown, are images, fluorescence images, of a particular protein, which is called Fib1, that they used as a marker for the nucleolus. Now, what's the nucleolus? The nucleolus touches on, one of the most important topics in all of biology, which is protein synthesis. So the nucleolus is the site, among other things, if I remember correctly, of ribosomal synthesis. And, if you stop and think about it, the energy budget of a cell, some huge fraction, and when I say huge, I mean like 80% or more, is dedicated to protein synthesis. And the key actors in this drama are the ribosomes, which, they engage with the mRNAs, they incorporate amino acids. For my purposes, one of the things I love about that is that they do this in a way that invokes proofreading, so there's error correction mechanisms. And so the nucleolus is the site of this ribosomal synthesis. That's all I'm going to say about it for now, but I encourage you to go look it up on Wiki and try to understand a little bit more about this key organelle. So what they did in this experiment is they measured the size of the nucleolus, and the way that they did that was basically to measure the fluorescence intensity of this protein Fib1. And so as the cells divided, you move from the position here. So this is early, and now time flows in this direction. The nuclear volume goes down over time. Why? Because the cells are dividing, and every time they divide, the whole system is at fixed volume, so every time they divide, each cell has a lower volume. And so what I want you to notice is this trend, the trend being that there's a scaling between the size of the nucleoli. If you'll let me, if you'll suspend your disbelief and permit me to say that Weber and Brangwyn were able to make the argument that the fluorescence intensity of this marker was a surrogate for the volume of the nucleoli. Okay, so anyway, there's this. So what I'm going to claim is that there's some sort of an interesting scaling, and that scaling is of the form that the size of the nucleolus is some slope times the volume. So this is scaling of nucleolus size m, which is for us is like a fluorescence intensity, versus nuclear volume. So that's observation number one. There's a second set of observations which are extremely cool and result in a different scaling. So in this case, using RNAi, they were able to do a knockdown that controlled the size of the embryos and thereby the size of the cells. And they assert that the total number of nucleolar proteins per cell is the same in all these different scenarios at the 8-cell stage. So now what they're showing us is a different scaling. In this case, the scaling is that as the volume increases, the nucleolar size goes down, and you can think of that as resulting from the fact that the concentration of nucleolar proteins is going down as the cells get larger, right? Because the total number of nucleolar proteins, the Fib1, is fixed. Okay, so those are our observations. So somehow, let's see, this one is some sort of a relationship of the form m is equal to, some constant minus some slope, some different slope times v. In other words, this is an inverse scaling, a different scaling relationship. So what I want to do now is I want to see if we can write our first concrete model down where we try to mathematicize this cartoon. And so conceptually, what I want to do is I want to write a rate equation. And the way I intend to do that, I'm going to repeat over and over through the term, and that is I'm going to say that the size of the nucleolus at time t plus delta t is equal to the size of the nucleolus a moment earlier. So this is size of nucleolus at time t plus delta t. This is the size of the nucleolus at time t. And now what we need to do is we need to figure out what are the ways that we can change the size. So, well, one thing we can do is we can add new molecules onto the nucleolus with a rate k on. And so I'm going to write that term in the form k on, and then n minus m divided by v, and then minus k off. So what's up with this? So first of all, and I need to multiply these each by a delta t. So just bear with me. So first of all, what is this stuff right here? So I should have defined n. N is the total number of Fib1 in the nucleus. M is the number of Fib1s in the nucleolus. So this whole quantity, which maybe I'll label in a different color, this whole quantity is the concentration of free Fib1, right? So the only Fib1s that can come onto the nucleolus have to be free. They can't be already part of the nucleolus, otherwise they can't. If they were, they're not able to do the dynamics and hop on. So, So that's what's going on with this first term. And then I remind you that the on rate, I'm going to use a notation. So we have an ambiguous notation we have to deal with. And the ambiguous notation is these square brackets do double duty. Sometimes they mean concentration of, but sometimes they mean dimensions of. And so in this case, the dimensions are inverse molar, inverse seconds. So meaning that the on rate, let's just look at the units. So we know that m is number. So we better make sure that we end up with units of number. So how do we reconcile that? Well, k on times delta t, according to this prescription, has units of inverse molar, And then I multiply it by n minus m over v, which has units of concentration. So concentration times 1 over concentration leaves me with no units left. It's units of number of. So that's what we observe. And k off has units of inverse seconds. So, that is our dynamical rule. And at the level of Python code or MATLAB code, this is what we would actually do. This corresponds to what's known as a forward Euler integration, where we have a first order ordinary differential equation that describes the dynamics of the nucleolar size, and we can solve this by time stepping with incremental small time step delta t. And I'm not going to get into the niceties of convergence of these things and what time step you need to use and so on. I'm going to urge you to use empiricism while being cognizant of the fact that there are subtleties. You have to be careful in the end. Now, I just want to note that I could always rewrite this in the form dm dt is equal to k on, and then times n minus m divided by v minus k off. So this is the differential equation version of that same law of time evolution. And we will interest ourselves in steady state. So at steady state, what do we know? We know that dm by dt is equal to 0. That implies then that, how do I want to think about this? So I guess I could say, well, let me unpack it a little bit. So first of all, this implies that k on times n minus m over v is equal to k, is equal to k off I don't know, let's instead write it like this, minus k off equals 0. I need to add another page here. Add page. Okay, and so I can think this through. This implies then that I have that n minus m minus k off divided by k on times v is equal to 0. Or m is equal to n minus k off. Did I make a mistake here? Over k on times v. No, I guess that's fine. Yeah, so what do I want to do here? Just bear with me for a second. I need to think, and I think I've maybe gotten myself a little bit confused, so I apologize, but just hold on a sec. So I start with the differential equation. I set the rate dm dt equals 0. That leaves me with this equation. I multiply by v and divide by k on. That leaves me with this. Yeah, and I can, as a last step, I can write this as n over v minus k off divided by k on times v. Now, observe that C tot, the total concentration of Fib1, is n over v. And secondly, notice that maybe what we'll call C star is equal to k off over k on. Why? Let's check units. So this is 1 over seconds, and this is 1 over molar, 1 over seconds, which leaves us with molars. So C star. So basically what we have is a simple relationship, which is m is equal to C tot minus C star C star times V. So that's a very nice relationship. And my claim is that this is the relationship that we want to use to explain this data. This is M is proportional to V. And we found with a slope, actually, because the slope is offered by this thing in parentheses, which is C tote minus C star over V. All right. Now, alternatively, we could have left things in this format. Oops, sorry. In this format. Because this is the one that we would want to use in the context of fixed n. So now it's n that's fixed, and what we have is we have m is equal to n minus C star times V. So this one, this case that I already wrote, is the fixed concentration scenario. And then now, in the fixed number scenario, we have this. So this is the fixed number scenario. So how do we square this with the data? So here, what Hernan Garcia did, book co-author on physical biology of the cell, is he said, well, look, this is cool. Why don't I use this data to pin down both C star and the n, which is the y-intercept. Okay, look at this equation. So given this equation, we can go down to this data, and we note that when V is 0, the y-intercept is n, so we can determine n from the data. And then the slope is minus C star. So those two quantities can be extracted from the data. Then that is cool because it renders this thing a prediction. No free parameters. So here we know that the line has a slope of n over V minus C star. We know n from the left-hand side. We've already determined n, and we can determine the total concentration by using the initial cell volume. And so we've got n over V, and C star was the slope from the left-hand side. Plugging those two things in leads to this line, which is the constant concentration prediction of this little toy model that I ran by you in this vignette. So that's basically the story as I want to present it, and actually in the homework, the students will have a chance to actually do this fit and work through the details for yourselves. But that's basically what I wanted to